All right, good morning. We're going to talk about cell division and reproduction. So we're going to talk about how one cell goes to two cells. We need that in order for reproduction, repairing injuries, growing, some of the basics we're going to go into with cells growing larger and then dividing into two cells. There are two types of reproduction. We're going to do both. We're going to talk about asexual and sexual. Asexual is where the original cell divides and makes a copy of its own DNA and then forms two identical daughter cells, versus sexual, which is where you need two parents and each parental cell is going to donate half of its DNA, in which then we will get an offspring that is a combination of both parents. Now, in prokaryotes, we're going to call that is our bacteria and archaea. We're going to call that binary fission. It is much simpler than what we're going to be going into with the eukaryotic cells. Prokaryotic cells have a single circular DNA molecule, so they just have to replicate that one circular chromosome in order to put one identical copy in each of the two cells that it will divide into. So if we can see here in our diagram here, we have a prokaryotic cell. It's circular DNA. We're going to make a copy. We're going to elongate our cell, pinch it in the middle, and now we have two identical daughter cells. So eukaryotic cells aren't that easy because they have multiple chromosomes. They are a lot larger than prokaryotic cells, and like I said, here we have multiple chromosomes, and they also have organelles and stuff that we have to be concerned about. So first thing, a little bit of vocabulary. So when we have DNA, when it's unwound, we call that chromatin. Now, DNA needs to be unwound during certain phases of its cycle, and that is the only time we can actually make proteins, is when the DNA is unwound so we can read it. And that's the way it is most of the time. However, if the cell is then told that it's going to end up dividing, it needs to wrap that DNA up so it doesn't get damaged in this process. And so it'll wind the DNA up into what we call chromosomes. So here's a picture of our unwound DNA, chromatin, and our wound up DNA, chromosomes. So here is a picture. Again, we're going to have our DNA, and we're going to get more into DNA replication in chapter 10. But this is called a monad, one copy. We really don't ever see, other than when we split these up momentarily during mitosis and meiosis, but you generally don't see a monad by itself. You will see a dyad. Dyad means it's two copies. That's that traditional X chromosome that we represent. We have, how we draw all our chromosomes as little X's, and then we have this central part called the centromere that's going to hold these copies together. These copies are identical. We call them sister chromatids. So what we're going to do is we're going to replicate our DNA so that we have two copies here, one for each cell. Before the cell divides, we have what's called the cell cycle. The cell cycle is divided up into two main phases called interphase and the mitotic phase. Interphase is broken up into three additional, you could say, subsets or subphases, G1, S, and G2, where mitosis is also kind of divided into two parts. Main part, which is the mitosis, where we're dividing, but more importantly, we're dividing the nuclear contents, versus cytokinesis, which is the division of the cytoplasm. So needless to say, when we get done with mitosis, this is also going to be asexual cell reproduction, and so the two identical cells are also called daughter cells. Here is that cell cycle. So we can see G1. G1 is where it's now called growth 1 and growth 2. We used to call it gap because we didn't know what was going on in those two phases. But we know now that once we get out of our mitotic phase, the cells are a lot smaller. So in the first gap, they're going to grow. They actually spend most of their time, if not all their time, in G1. Not all cells divide, but this is where they're doing its work, the job that that cell is meant to do. It will only advance into synthesis if it is told it needs to divide. In synthesis, it will replicate the DNA. And then we have our second gap, which is basically the cell preparing to divide. And then we go into mitosis and cytokinesis. So here is, again, our unduplicated chromosome. We're going to make a copy in synthesis so that we can put one copy in each cell. Now, normally I would draw out mitosis on the board in this format. Of course, I can't do that, but I did put videos in the module, which shows me doing these on whiteboards in my classroom several years ago. So you do have those to help. So I'm just going to kind of go through the diagram really fast. So we have interphase, and again, in interphase, the DNA is not wound up. So we have chromatin, we have these organelles we really haven't talked about much called centrioles, and they're going to aid in dividing up the chromosomes. We have, we used to use that, an acronym like PMET, prophase, metaphase, anaphase, telophase, but they decided to throw in prometaphase. There's a lot going on between prophase and metaphase. Prometaphase is kind of like in the middle of that transition. So in prophase, we're going to do a couple of things. We're going to wind up our DNA, which takes time. We're going to wind them up into chromosomes. And then the next thing we're going to do is we're going to dissolve that nuclear membrane here. As we start to transition towards metaphase, we will then line up in the middle. So when the chromosomes are all lined up in the middle, that is called metaphase. So always remember middle metaphase. And then anaphase, we're going to pull those sisters apart. So always remember anaphase apart, AA, anaphase apart. So we're going to split those sisters. They're going to be attached to these spindle fibers, and then those spindle fibers are going to actually kind of pull the sisters apart and break that duplicated chromosome here. Duplicated meaning it's got two copies. We're going to split our copies up, and then we're going to pinch in the middle of an animal cell here, and then we are going to start to divide up the contents. So right now we're dividing up the nuclear contents. Once we're done, we're going to start forming our nuclear envelope, but we also have to divide the cytoplasmic contents, and that is basically the organelles that are in the cell. So that is what cytokinesis is splitting up those organs. So we make sure that those organelles go. We get smooth ER, rough ER, mitochondria in both of those daughter cells. Now, like I said, in animal cells, this is easy to do. We have what's called a cleavage furrow. We have this contractile ring, and basically it kind of pulls closed and then pulls and then divides those two cells. But it's not so easy to do this with plant cells, and that is because of the cell wall. So in plants, we're going to form what's called a cell plate. It's going to go right down the middle of the plant cell and split it into two, and then eventually those two cells will enlarge to become the full size that they need to be. All right, so a little bit different. We don't have that cleavage furrow in plant cells because of the cell wall. We also have these things called anchorage, cell density, and chemical growth factors, and that is things that keep cells in check. We don't want cells to grow out of control. So we have these growth factors, we have this thing called density dependent inhibition, and we have anchorage dependence. So I'm going to kind of show you that in the next few slides here. So this is anchorage. So the bottom of these dishes, these are special dishes we use in cell and tissue culture when we want to grow eukaryotic cells. They are specifically mammalian cells. They are etched on the bottom, so it allows the cells to actually anchor. And once they're anchored, they can start undergoing mitosis. They will undergo mitosis until they are touched by other cells. That's that contact inhibition. Once they have contact with other cells, they will stop dividing because we don't want like a tumor to form. So we can take out a couple cells and they'll undergo mitosis and fill it nicely in if these cells are functioning properly. If they are not, then, well, that's where we're going to end up with things like cancer. So there are three major checkpoints that cells have to have during the cell cycle to make sure that everything is healthy with that particular cell so that it does not form into a cell that would hurt that organism. So we have these different checkpoints here. So we have our checkpoint in G1. Again, we're going to make sure most of our cells will actually sit here because a lot of our cells will never ever divide. Matter of fact, we have this thing called G0, where a lot of your cells will even exit the cell cycle and sit in this static phase where they'll never divide. If they're given the green light, they move into synthesis, they go into gap 2. Before we go into mitosis, there is actually a proofreading process to make sure the DNA did correctly replicate itself. If there are errors in the DNA replication at this point, the cell will actually self-destruct. Okay, it's called apoptosis. If we do get the green light, we'll go into mitosis. We also have, notice a little check here towards the end of mitosis. If things are not dividing properly, again, we can undergo apoptosis there. So these are all different mechanisms that the cell will use to prevent things from going bad. They either fix the problem or self-destruct. Cancer cells are cells that have gotten through, where these checkpoints haven't worked properly. You guys know what tumors are. So this is where not only does the tumor, again, our immune system can find some of these rogue cells and kill them, and that's great. But other cells that have multiple problems with them, they don't have that anchorage dependency, so they divide without being anchored somewhere. They grow without that contact inhibition. Those things cause tumors. We have different types of tumors. We have benign tumors that are tumors that just stay in their site and don't really cause any problem. And then we have malignant tumors that will actually spread to other parts of the body. We call those when the doctor says you don't want to hear that word, that it's metastasized. That means it has spread. And this is just a duplicate slide. We do name cancers according to the organ or the tissue type that they originated in. So carcinomas are generally cancers that have to do with like your skin, the external or internal body coverings. Sarcomas are supportive and connective tissue, and lymphomas, leukemias are your blood forming cells. All right, so I am going to pause here because I kind of want you guys to now go back through and read the stuff on mitosis, possibly do the mitosis lab, and then come back and we'll go into meiosis, which is the second process in this chapter. But I want to make sure you have mitosis first before you continue on.